Great. So I just want to make sure everybody can see our agenda here as we're talking. Just wanted to verify that. We went through the quick poll. You're going to meet your presenters, and then we're going to introduce you to the GOT protein kit and how it can be used to measure protein content from both animal and plant milks. This is a lab that we use or do with Bio-Rad. It's a great lab. Then we'll have a hands-on demonstration with Vernier spectral analysis. I might show you graphical analysis, some cool stuff that you can do there. And then we'll have a Q&A session. All right, so we've done the audience survey questions. So I'll let Sarah introduce herself.
Hi, I'm Sarah Talarovic. I'm a biology staff scientist for the Vernier, and I taught college for about 15 years, all different sorts of biology classes up through things like neuroscience, and that's me.
Hi, everybody. My name is Damon Tai. I'm Bio-Rad's curriculum and training specialist for the Western region of the United States. I used to be a high school teacher up in Portland. I worked on the Human Genome Project and a bunch of large sequencing projects for about 10 years, and then moved over to Bio-Rad a little over 10 years ago to help drive more hands-on biotechnology education into classrooms.
Hi, and I'm John Melville. I got my bachelor's degree in psychology and biology at Sonoma State. I have a PhD in zoology from Oregon State University, same place that Sarah went. And then I did a postdoctoral fellowship at the University of Oregon. Both are in neuroscience. What's most important, though, is that I was an associate professor of biology at Wartburg College, and I also taught at a small two-year college in Oregon called Lane Community College. My background is in majors and non-majors courses in biology, pretty much everything from neuroscience, physiology, general biology, which this would be great for, even marine biology. But in almost all of my courses, I had a real focus in active learning, and then an emphasis on data collection and inquiry, and also research methods and research. So definitely focusing on hands-on. And that's what we're going to talk about today. So just remember, go ahead and ask all your questions in the QA. Someone will get to them, or will respond, or will tell me. They'll just say, hey, John, there's a question. No, I think even someone put something in chat just now. Someone will be answering your questions there as we go, and if time allows, we will discuss the questions at the end of the talk. You might want to find spectral analysis right now if you just want to take a look at it. It's on our website. You can search for spectral analysis. Download it if you don't already have it. It's great. It's free. It's to be used with spectrometers, and you can analyze data with built-in analysis tools. And it's going to be what I'm going to be using today when we talk about this lab activity. So once again, we're going to have a live demonstration of experiment 5 from our great Investigating Biology Through Inquiry book. And in this experiment, we use a GoDirect SpectroVis Plus. That is an array spectrometer. So it kind of is like a Spec 20, only better. It captures an entire array spectrum. So you can see the entire wavelength spectrum. And then you can then begin to do an experiment. And what's also great about it is it uses a Bio-Rad kit. Bio-Rad has several amazing, excellent kits that we use in a lot of our curriculum. And they're just wonderful to use and incredibly popular. And Damon will be talking about the kit itself.
So the
go
kit
ahead,
we're going to
Damon.
use today is the Bio-Rad GOT protein kit, which is a Bradford assay. This kit, most Bio-Rad kits are built to serve kind of 8 workstations and a full classroom. This one serves a ton of people just because this is the smallest Bradford bottle we can get you. So this one can serve up to 320 students instead of just the normal 32. And at 189, that means that each workstation ends up being a relatively small amount for the price. If we dig into the kit a little bit more on the next slide, at its core, what's going on is you're going to take Bradford reagent, i.e. Comasi, and this is a compound that when it binds to a protein, it will basically do a spectrum shift of where it's absorbing. So you'll basically, John's got visuals that he'll show you today. You'll basically shift from kind of a brown colored reagent to when it complexes with protein, it'll shift into kind of a brown looking reagent. And you can monitor this with a spectrophotometer. And that's what we'll be doing today. So go ahead and go to the next slide. With that, the spectrum you're able to collect, you're then able to use Beer's law to understand the concentration of the protein that is in the solution. So you can literally go after any sort of material that you can get aqueous that has protein in it and figure out what the concentration of protein in there is. So Beer's law, if you're not familiar, it's just the absorbance equals the molar absorbativity times the path length of the sample, and then multiply that by the concentration of the compound in solution. This usually gives you a nice kind of linear fit, and so that way you can take unknowns, plug them in to a nice linear fit curve, and be able to figure out exactly what that unknown has.
Great. So once again, we're going to be using that kit in this activity. This lab is actually in our Investigating Biology with Inquiry book, number 5. And it really is just an activity where students are going to be looking at, they make a standard curve, they measure an unknown, and then they use inquiry, which means you have them go out and they just try to figure out, well, what is the protein content of some compound? And so a lot of what we're going to talk about is what are different avenues for inquiry, but also I'm going to show you how the standard curve is made, how to measure the unknown, basically do what we call the preliminary activity. And then I'll talk about lots and lots and lots of different things that we've done. Just to let you know, Vernier software and technology is for teachers by teachers. Everyone at Vernier that you're talking to has been a teacher, was a teacher. We like working with teachers. That's our focus. So I always like to say Vernier is for teachers by teachers. We have chat on our website. You can always call. If you ever need to get in touch with me or Sarah or the biology team, you can always email biology@vernier.com and someone will get to you right away. We're going to be using spectral analysis software, which is live. Oh, I'm going to be doing it live, but it's also free. But I just wanted to let you know there's a folder that you have access to that Angie gave you. And I have put in there the student instructions for this activity for LabQuest and also LoggerPro. So we have different software titles that we've used. I love LoggerPro. I've used LoggerPro for a long time. Our LabQuest is a wonderful device, and we have that as well. And so I put all 3 types of student instructions there. I'm going to be using the spectral analysis ones, but I put those there. The most important part of our inquiry biology book is probably the instructor notes. The instructor notes are what tell you how to do the lab, what are the ideas for inquiry, what are all of the different things that you can do. I'll give you I'll sort of show you how that looks here. Let me just show you here really quick. You're going to be seeing this display over here. This is my spectrometer and my materials here. This right here is the instructor information. It's got the learning outcomes, correlations, results for the preliminary activity, but really important sample data, lots of sample data. So that's like one activity that you could do. It's got other activities. It's got data for lots of different experiments. And it also tells you, hey, what are the experiments that I shouldn't do as well, which is really important. The preliminary investigation looks like this, right? So this is the basic information for it. You have access to this in the folder. The other great thing about our book, the book is that it contains the digital contents for the student and instructor versions, and you can edit them, and they're in a Word document form, so you can modify them, change them if you need. Now, this lab, when we originally wrote it, was pretty much focused on investigating animal milks, because we developed it with Bio-Rad. Well, they had it out there, and we put it in our book about 10 years ago, I think, over 10 years ago. And there really weren't a lot of plant milks around. It really was just things like high protein drinks, right? I think we developed it with Don Volz, who was in Germany at the time, and he was like, hey, I found this thing called rice milk. And I was like, sure, go ahead and measure it. Well, today, there are all sorts of different types of milks and high protein shakes that are on the market. I have a collection right back here where, it used to be years ago, there were really only 3 things: whole milk, reduced fat milk, 0% milk, and then high protein drinks. I also have a Rebel drink here, which is like a natural high protein drink, but there has been a dramatic increase in just plant-based milks. right? A range for putting in your coffee is oat milk. There's almond milk. There's soy milk. There's goat milk that you can buy now. There's all sorts of different types of milk. So our idea with this demonstration was, hey, wouldn't it be great if we could analyze and look at all of those different milks? And the answer is you can. So once again, we're just going to compare protein content on the label to the measured value of these different types of milks that you could buy. It's super simple and easy to do. This kit from Bio-Rad is great, simple and easy to do. And I wanted to just let you know that it can be done in a qualitative or quantitative way. If you're teaching college, it's great that it's quantitative. You can create a standard curve, find your unknown, and calculate the total protein content, right, that you have. But if you were doing an intro level biology lab and you just wanted to show students, hey, there's differences, you could just show them the difference in concentration, the different absorbance values, and show them that absorbance values are higher or lower. In fact, what's great about this assay is it's colorimetric, so you can easily see things with high protein are turned blue, things with low protein content are just a brownish color. And I'll show you that too. One thing just to specify with our spectrometer is you want to make sure all the readings are bracketed for best reading between 0.1 and 1 absorbance. For this kit, the absorbances goes a little bit high to like 1.2 to 1.5. That's okay, but you'll see a little bit of nonlinearity in that range. It's fine for this activity. You could dilute if you wanted to, get better results and make it more linear, but it's not required. The last thing that I wanted to show you before we go to the live demo is down here. I created a simple nut milk procedure because people have been making nut milks. Sarah could tell us this, or she just did, that they've been creating nut milk since, I don't know, hundreds and hundreds and hundreds of years, and different types of nut flours. And creating a nut milk is actually quite simple. Let me show you an example of what you would do here. We can talk about that here. You take a source of nut. Here's some almonds. You pour them in water. You would pour this in a certain known amount of water, let it sit, add more water, grind it up, and then basically run it through cheesecloth. And after you've run it through cheesecloth, you get this foamy kind of mixture here of ground up whole almonds, right, with water. It's just whole almonds with water. And then you run that through cheesecloth, and you get something that looks a lot like milk. So we're also going to measure the content of almond milk. This is almond milk. And then I got some fresh pecans, and this is pecan milk. And I've prepped them the same way. I've taken 100 grams of nut. I've added it to 100 grams of water. I let it sit overnight. Then I add another 200 mils of water. Then I grind it up, run it through cheesecloth, and then I just measure the absorbance reading. All right, any questions before I go to what we're going to do next is I'm going to switch over, and I'm mostly going to just be showing you video. So if you have any questions, now would be a good time to ask them as I move over to showing you how we actually do the lab and run spectral analysis and use the spectrometer.
And feel free, everyone. You can certainly ask questions as we go. Just put them into the Q&A, and I would be happy to relay them to John as he goes.
Well, that was a good question. Someone had a good question.
Oh, it looks like there's one in the chat.
Wouldn't
different proteins cause differences in absorbance?
Damon, do you want to take that one?
That's a really good question. So depending upon the amino acid content, so which amino acids are there, you might see a slight shift in how the Bradford assay quantifies how much protein is there. Because it's, if I remember right, it is mainly interacting with the hydrophobic amino acids. And so if you had something that was really devoid of the hydrophobic amino acids, you might not detect that there's much protein there. But for the most part, once you get to a certain size, most proteins have a normalized kind of amount of these hydrophobic amino acids.
Yeah, I think we also have instructions in the instructor notes or in one of our labs on how to do that's a different protein assay that's dependent on secondary structure. I'm forgetting the actual name of it, but I know it's in there, but that's actually pretty neat to show too. But what I've found is that you get pretty consistent good values, but yes, the protein content can vary. The Bradford is specific to a given amino acid, so different amino acid concentrations could change it, but you're still measuring it relatively. And that's another good question to remember when we come up to making the standards. So let's switch over and talk about the standards, because that's another
Actually, we have
question.
one more quick question.
Good.
And it's good timing for this one as well. Besides milks, what are some other proteins that can be quantified? Can you do something like eggs with albumin, for instance?
I think it's pretty much, I mean, Damon is better at answering this, but anything that you can get into aqueous solution, you can probably measure.
Yep, that's right. Basically, anything you can get into solution, you can measure. Just the big thing is you want to filter it well, right? Depending upon what that substance is that you're trying to get into solution, so that way you don't have a bunch of things scattering light when it's in there.
I mean, this gets back to an important thing before we move on, maybe that Damon, you might want to address, but it's one of the things that I really love about Bio-Rad, right, is the Bradford assay is a standard assay that everybody uses. Correct me if I'm wrong, Damon, but this is a standard assay in biotech. And the reagents that you're getting from Bio-Rad are the same reagents that someone would be getting if they were in a lab that's doing, the Bradford assay. So like when you get your Bradford bottle, I mean, I don't have it here. I mean, I've diluted my Bradford assay out here. Yeah, there you go. Damon's got it.
There's the big leader of it. Yeah.
Yeah, I think that's pretty much the same bottle you would get if you were at a biotech company and you ordered it. So
yep, it's the same one. So with a lot of the Bio-Rad kits, what you're getting is stuff that's normally going out to researchers, but you're getting access to it as an educator. And so if you go to Bio-Rad's website and you type in a Bradford assay, what you'll find is this same bottle being sold to groups that are doing food monitoring and things like that, with the associated standards, which is also what comes in the kit. And those standards are really one of the things that's like really, really hard to build and to get a really nice, clean setup. And Bio-Rad's really well known for making standards throughout the industry. And so these are a set of BSA standards. And just one caveat, that is one of the things that when it shows up, you want to make sure that that makes its way into the fridge, that those proteins don't degrade at all.
Yes, the whole kit needs to be refrigerated.
Damon, we have a quick question. What's the shelf life of the Bradford?
If you store this stuff at 4 degrees, I mean, the Bradford reagent will last, Bio-Rad guarantees everything for a year, but like real shelf life on a Bradford reagent is closer to like 3 years if you're keeping it at 4 degrees.
And I just verified that today. So I use the reagents that we've had when I came, and I used the reagents that have been sitting in the fridge for almost exactly a year, and I got good data. So they should be fine. What I wanted to show you, what's really cool, I'm going to focus here over, just let me know if you can't see the spectrometer here and the camera. What I want to show you is this is the set of standards that comes with the kit. And what's really great is it comes with 2 full sets of standards. So you see right here, this says 2 milligrams per mil. This is one standard, but you see I don't have a star by it. That's because I haven't opened it. So what's really nice is it comes with like a whole set, I believe, of 7 standards. 1, 2, 3, 4, 5. So yes, there's 2, 1, 1.25, 0.5, there we go, and 1.5. So we've got 2, 1.5, 1, 0.5, 0.75, 0.75, 0.125, and 0.25. So 7 standards. And this is enough for an entire classroom. Like you're only used for each student is only going to use, if you were going to make up your own standard curve, you would only use 20 microliters. That's what's super nice about this lab. So let me I'll show you an example of that right here. Is if I put these back over here, let me grab my standards. So here's the 2 mil standard. You can see it's this brilliant blue. Can everybody see that? How blue that is? I can bring it up, or I can bring this down. That's very blue. 0.25, much darker brownish color. All right. Let's move back up. So what I've done here is I've just created all of these standards where I've taken 1 mil of Bradford reagent and I've added 20 microliters of each standard to create a set of 7 standards. And that's what the first activity does, is you actually just create a standard curve. So I have already created my set of standards. And then you also create one unknown for the students. I have that here. I have one unknown. This is, let's see, reduced milk fat right here. And then let me just go back and I'll show you how to use the spectrometer. And then don't worry, we'll come back, or I can show you what it actually looks like. I mean, to make the Bradford reagent, it's quite simple. There's the color right there. Let's just zoom up really quick. I think we've got time. There we go. So you can see it's a nice gray color. I'm just going to take 20 microliters of, let's see, microliters of the 2. I'm going to add this. And you should see at the top, oh, I dropped that, hope that's fine. Blue begins to form. It's not reflected too much. You can see it's blue. Luckily, I have a little cap. So our cuvettes come with a little cap. And then I can just shake this up and you can see it's turned a blue color. That's what the assay is. Different shades of blue. All right. I'll zoom back out. So what I'm going to do next is I'm going to focus on using the spectrometer. This is our little array spectrometer. I've got it plugged into USB. It also can be used via Bluetooth. That's what this little button is, but I'm not going to use it on Bluetooth today. It runs on Mac, PC, iOS. You can even use your phone if you wanted to, and Chromebook and Android. The light path goes this way. So there's a lamp here and a slit here. So if I take my blank, this is my blank here, you can see there's the clear path. This side isn't ridged, or it's a 1 centimeter path length is this way. That means I want to put it in like so. So the light goes through that way. All right, so let me launch spectral analysis. Once again, let me know if you can't see anything or if I'm going too fast. I'm going to relaunch spectral analysis here. And I'm just going to collect a wavelength first just to show you what it can do, and then I'm going to show you how to create your own standard curve. So let me I'm going to recalibrate the device just because it's been sitting here for a while. Normally, when you calibrate the spectrometer, it needs to warm up for a while. This one's been warmed up for quite a while, but I'm just going to let it warm up. Now I can skip, finish calibration. There we go. Let me just put that here and I'll just try and minimize this a little bit. There we go. So now I'm going to put in the 2. That's a dark blue. toss it in there. Let me collect a spectrum. Great, so there's a nice spectrum. And you can see there's this nice peak that we can track. So let me go to new experiment. Because what I'm actually going to do is I'm going to collect an entire series. So what I want to do is create a standard curve. So I'm going to do versus concentration or Beer's law. So remember that spike that I just showed you, that increase in absorbance, that's being shown right here. I have the same sample in over here. If you look, I've got the same sample in, and it's got a peak. And so then all that I'm going to do is I'm going to minimize this because I'm just going to be putting in samples back and forth, and we're going to be measuring them. And I don't think you need to see my hands over and over. So I'm going to minimize that. And the first thing that we're going to do is we're going to pick a wavelength. Well, looks like there's a peak right around here at 590 or 593. The Bio-Rad kit tells you to use 595, but anything in that range is going to be fine. So I'm just going to pick 593. I'm going to hit done. What's great about how our software works now is that it's going to allow me to collect a set of standards or create my own standard curve. So I'm going to hit collect. I want to make sure that this reading down here is stable. It says it's 1.391, 1.384, not moving. And hit keep. And then it's going to say, hey, what's the concentration here? I'm going to type in 2. Now, don't worry that it says moles per liter. The chemists want everything in moles per liter. I'm going to show you how to change that later. So don't worry, you can change that. So I'm going to say keep that point. Okay, now I'm going to take out the 2 and I'm going to put in the 1.5. We're going to wait for this value to stabilize. So if any of you have used a Spec 20 before, right, you would dial in your wavelength. That's all that we've done. We're putting in our sample, but our software is picking the data point for you and then creating the actual curve so that your students don't have to create that standard curve. Don't worry, you can have them create it by hand later if you want to as well. Just ask. So I'm going to hit keep. And I know that this is 1.5. So I'm going to say keep that point. And then all that I'm going to do is I'm just going to continue to create this standard curve. I'm going to put in 1. So right, I know what these standards are. I created them. I took the standards, I reacted them, I've let them sit. All right, that's good. 0.6, 0.62 looks good. Is it stable? That's 1. .75. If anybody has any questions, go ahead and ask. .75. There we go. Put in 0.5. Make sure this is stable. Could this be done with a GoDirect colorimeter? Yes, you could use a GoDirect colorimeter. You would have to use graphical analysis, but that's a great question. That is actually in the teacher's notes that you could use a colorimeter with graphical analysis. You would have to pick the wavelength that is closest though. But you could. We don't have the specific instructions written for colorimeter in this lab book, but it would be very simple and easy to do. If you want to do that, just let me know, but you can do the Bradford with the GoDirect colorimeter. One of the nicest things also about the Bio-Rad kit is it comes with semi-micro cuvettes. So the semi-micro cuvettes hold 1.5 liters, 1.5 milliliters instead of 3 mils. Yeah, thanks, Damon. I knew you were going to laugh at that. And it's really nice because you get like a full set of over 100 of these, which are great as well. Vernier sells standard cuvettes, which are 1 mil.
And
John, we have
Bio-Rad
a
says these
question.
are, I was gonna say, and Bio-Rad says they're disposable, but you can really, if you kind of keep, keep them clean, you can use these for a couple of years before they start to accumulate mini scratches. So I would keep that foam rack and I would just re-rack the stuff after I use it every year.
Oh yeah, I was going to say I've done this lab for almost 10 years, and how I clean them afterwards, the Bradford reagent is I just put a little bit of bleach in like 5 liters of water, like just several drops, throw my cuvettes in there, let them sit overnight, and then just wash them over and over. And I've used them for years that way.
John, we have a question from the Q&A. What are the units of the protein concentration of the standards?
Don't worry, I'll get to that. The issue is that right in our software, concentration, you can't change it to start off, but I'm going to show you that how we're going to actually change that. That is a great question. And then I'm just going to put my, so I think that's good. I'm going to stop. Thank you so much for waiting. That was like actually a plant that was in the audience there, because that's exactly the next thing that I'm going to do. So I'm going to hit stop. I've created my standard curve. Does everybody see that this is like relatively straight? It's a nice standard curve there. So what I'm going to do is I'm going to tap on the bottom left corner where it says graph options. And I'm going to say apply curve fit. And I mean, this curve fit could be better. These little points are a little bit low, but hey, you know what? That's okay for me. What I found when I did this lab is that my pipetting is rusty. And I've also did a serial dilution and got good data. So, this is a nice linear fit. So the next question you said is like, well, what are the units, right? What are the units of the standards? Well, the standards are in units of milligrams per mil, not moles per liter. So to change that, I'm just going to tap up here where it says column options. And I'm going to change this to, I'm going to leave this concentration, but I'm going to change this to milligrams per mil. And now we should all be happy, or happier. So now if I hit under curve fit, if I go to interpolate, I can slide along this graph, right? And I can find out, this is my standard curve. I can find out the concentration of an unknown. So next, I'm going to take some non-fat milk powder, or not non-fat milk, but I'm going to take some reduced milk fat. Let me just show you which one that is. This one. This is just some reduced fat milk. Let's take a look at it. I'm going to put it in the spectrometer. We'll measure it. I'm just waiting for this data point right here. This is always live. So that's a key thing about all of our software is there's always going to be a meter. There's always going to be a data table. There's always going to be a graph. Whether it's video analysis, whether it's spectral analysis, whether it's graphical analysis, there are always those 3 components, and the meter is always live. So right here, it's like, okay, well, 0.436. Well, let's find 0.436. If I go over here and I go down to 0.436, or maybe 0.4. There we go. It's saying that's 0.66325 milligrams per mil. Write that down. Now, in this lab, these are all diluted by a factor of 50. So I just want to multiply that by a factor of 50 to find out what its concentration really is. So let me pull up a calculator and I can hit clear. And I can do 0.66325 times 50. Once again, this is all in the actual lab itself. It tells the students, hey, you need to multiply by 50 to get the proper concentration of your unknown. So it says it's 33. So 33 milligrams per milliliter. let's see here, protein. Now, the tricky thing there is then you would have to convert you'd have to know, well, if that's 33, I would then have to know what the serving size is, right? That's the actual measured amount. If you go back and look on the labels, I have right here, I went through and looked at all of these labels. I've got almond breeze, oat milk, soy milk, goat milk, rebel milk, muscle milk, non-fat, and reduced fat. The non-fat milk, which I believe that one is right there that I actually measured, says it's the protein concentration. If I take the protein on the label and I know the actual serving size here, it says, hey, the protein concentration on that should be 41. And the protein content that we measured was 33. So that's a little bit low, right? But that's how you could do this lab. And all I did, this is graphical analysis, and all that I did is I just created columns for the type, the protein on the label, the serving size on the label, and then I divided to get the protein concentration. So I could then compare measured versus reported. I'll move this over here. So what would we like to take a look at next? Let's take a look at, oh, pardon me. So that was just normal milk. Let's take a look at, here we go. This is some rebel milk. It says it's got a lot of protein. It says it's like got 16 grams of protein per serving. So let's see how high that protein is. So I'm going to put it back in the spec. Let me minimize the spectrometer. Let's put it in here. It says it's 0.55. So let's just wait. Let's put that curve fit back on there. Now remember, you could have your students write down this linear fit, right? And not have them do it this way. You could have them write down the linear fit parameters here. You've got the slope. That's really all that you need, 0.625. You got the intercept, everything else that you need. And you could just have them report this value and then actually use the formula to calculate it. And instructions on how to do that are actually in the lab as well. So you can make this as mathematically rigorous or as simple as you need it to be. So let's go find 0.579. There we go. So that's about 0.89382, 0.893. Let me find the calculator again. Did I get rid of my calculator? Let me find the calculator again. All right, so we can take 0.89382 times 50. Oh, it says it's 44. Let me look at my trusty table that I created. Its reported value is 66. So that's a little bit lower than it's reported, but it still is much higher than the non-fat milk that we got. So that's another thing that I could do here is I could say, hey, you know what? Let's create a column, add a manual column, and I'm going to call it measured. Right, and so I just have the nice table here. I know that when we looked at the first one, reduced fat, or the non-fat, that was reported at 33, and Rebel was reported at 44. And if you want me to show you how to set up graphical analysis to do this, it's pretty easy to do. I'm just kind of using it like Excel. I've just put in categorical data. I've put in a set of values. I've done a simple multiplication. And then what's nice is I can show measured next to the protein concentration. So right here's non-fat. You can see those are pretty close to each other. Rebel, once again, less, but close to each other. And you could have your students create a whole table like this where they measure different values. Okay, what about muscle milk? Let's take a look at muscle milk. And then we'll get at some plant milks because I know that Sarah has some really cool stuff to say. Here's the high protein. I'm going to put the high protein drink in there and we'll see how high protein it is. Oh wow, so that's a lot. That's 0.916. So you can go, let's see if it stabilizes. We can go up here. Let me do another, let's apply that curve fit again. And once again, just hit interpolate. There we go. So it says 0.925, 0.9. You could say it's right around there, about 0.9. It's bouncing a little bit. 0.8, 0.9. Let's just say about 0.9. That's 1.41. And then we have to multiply by 50 again. 1.41 times 50. So 70. Let's take a look at our little graph. Oh, we got 70. So they're all measuring a little bit lower than they should. Oh, well. But you can see the trend is still the same. The muscle milk has the most protein relative to all the other ones. Questions so far? Oh, by the way, if you don't like these colors, you can change them. I could make this, let's make this color red. There we go, but better contrast. Now, those tables that I'm showing you, that tabular format, that does require a subscription that's called Graphical Analysis Pro to be able to make it do that and to do some calculations, but it's very affordable. Let's see. All right, let's go back here and let's take a look at, let me show you the video. This actually will be pretty interesting, I hope. Let's just compare almond milk. So almond milk that you buy at the store to fresh almond milk that I just made. How does that sound? Any other questions or anything before we roll along? Does anybody have some other things they want to take a look at of all these milks that they've got there? But let me go back to the video and let me just show you what we have here. So if I go to the actual video, we find it here for you. And I'll zoom in here so you can see it nice and close. Okay, this is Almond Breeze. This is just the almond milk you buy in the store. This is fresh almond milk that I made. Hopefully you should see that those are totally different. And this is actually a really cool thing, that idea that Sarah came up with, which is, a lot of these milks that you buy, you would assume, even soy milk and other things, that they're actually high in protein. Well, they're mixed with other things, water and stuff like that, to create a milk-like substance. It doesn't mean what you buy in the store is going to have the same protein content as the actual nut or flour or whatever it is that you actually buy or use. And I just wanted to let you know that you don't have to stick to nuts. I've made lentil milk. I've made garbanzo bean milk. I've made pea seed milk. I've made different type. You can make any type of, once again, dry material. You can actually create a milk-like substance. Sarah has even, Dr. Tlarovic has even made flour milks and is able to measure those, if I remember correctly. Correct, Sarah?
Well, they weren't really intended to be milks. They were. We were actually wondering if you could just sort of proof of concept. Could we test the protein content of different grains? And we actually made a suspension of flour into some DI water, then followed the general procedure of adding it to the kit. And you can actually get a measurable difference in protein content. I had a selection of proteins that were reputed to have different levels of protein in them. And you can see differences. It's pretty neat. So you can kind of, if you want to get really outside the box with your students, anything that can be suspended in water, or create basically create a milk-like substance by putting it into water, you can try in this kit. So flours of different types, spelt versus traditional wheat versus artisan high gluten bread flour. We actually had some success with those. So
Yeah, it really is an amazing kit for inquiry. So I've just tossed in some almond breeze. That's at 0.117. And that's like off scale. Like I don't even know that I can calculate that. It's so low. And it's got about 1 gram. It's reported to have about 1 gram per serving. So very little protein. Let's just take a look at the fresh almond milk. .685, 0.7, let's see, 0.6. I mean, it's so much higher. And it's just a really, really simple activity to do with students. All that you need is some cheesecloth, water, grind it up, and measure it. By the way, there's another great kit that we've done with Bio-Rad, looks at solubiase, and you can do the same thing with mushrooms, where you grind them up and look at their enzymatic activity. And you could also use the Scott protein kit to look at the amount of protein per enzyme activity that's in it as well. So this kit can really be used in many different ways besides just this one as well. All right, so let's see, what else do I want to, what did I want to talk about here? Oh, pecan milk. So you can see this is the fresh almond milk. Let's take a look at some fresh pecan milk. It's got almost no protein in it whatsoever. What's nice is I bought these nuts from the store, and different nuts have different amounts of protein in them, and beans. Right, and you will see the same general trend when we've done this activity with like garbanzo beans, lentils, and almonds, and cashews. What you'll see is the protein content on the label of the actual nuts themselves does correlate. So the one that says it's got the highest protein content actually does have the highest protein content. All right. So let me back out of spectral analysis for a minute, unless people have any questions. Back out of this view. Oh, let's not do that. There we go. Minimize this. So I just wanted to reiterate, we're going to cover a few things, and then I can go back and show you anything that you want as we've talked through this so far. We've been using Vernier spectral analysis. It's free, doesn't cost anything. It's what you use with our spectrometers. There's this really great spot on our website, pardon me, that is the co-branded Bio-Rad Laboratories and Vernier software website. The reason why I think this is a great spot is because, let's say you don't have the book, but you want to do the activity anyway. The experiments are here along with the product that you would need to use. So we have both of these activities, the inquiry and non-inquiry, because these activities are also in another book that we have that are in a non-inquiry format. So you can do either of these activities. In this case right here, like for the Got Protein kit, if you want to just do a standard lab where you tell the students what to do, it's all preset. You can use macromolecules experiments with protein. That just looks at high protein drinks and regular cow's milk, or you can do the inquiry-based version. But all of the other kits that we have lab experiments with, whether they're in a book or they're on the website, the instructions are here. So if you want to study photosynthesis and respiration, it's there. For proteomics, we have all of those. PGLOW, sadly, but hopefully sometime soon, we still have these kits that are here. We do not have experiments designed for them, but we still have products that would be useful to be used with them. And then just once again, I just wanted to let you know that what I'm talking about, so far what we've been talking about is this lab book, the Investigating Biology Through Inquiry lab book, which is a great resource. I'm a little bit biased because I helped develop and write it. I think it's great. And Don Volz, who also helped, and Mike and a bunch of other people. But I also really want to give a shout out to Bio-Rad. When we were developing this a book, they were instrumental in developing several of the labs and activities that are in it. So we're really work as a great team, I think. All right, so if you have any questions or anything you want to talk about, go ahead and put them in the Q&A right now. Don't forget, you can go to vernier.com. You can email support@Vernier. If you want to get to the biology team, you can email biology@vernier.com. And if you want to get if they want to get a hold of you, Damon, what do they do?
I can drop my information in the chat for you guys right now.
Sweet. And if you want me to, I can also very easily show you how to set up that file that I did in graphical analysis and how to create a file that shows categorical data. So if you actually want to see that right here, like how I created this file, I could show you that. Or if you want me to go back through and show you how to use spectral analysis, we can do that as well. In addition to collecting just individual spectra, spectral analysis can create a standard curve, but it also can be used for enzymatic analysis. So you can track a wavelength as a function of time, which is also another great way to use it.
We had a quick question from someone, which was, will the video be available to use to rewatch? And yes, it will. The video, I'm not sure exactly when it will be posted, but I know that we are being recorded, and we do have plans to put that onto the website for anybody to rewatch, or for anybody who missed it. So feel free to, when the link is up, feel free to share it with colleagues. Usually we have them up within about, I want to say definitely within the week.
Yeah, another great thing just to remind everybody here is that Sarah and I and the biology team, we can do a free demo for you if it's under an hour. It's really easy for us to show you how to use a spectrometer, show you how to do something. If you have any questions, please just call, get on chat, or just email biology@vernier.com and we'll get to you. We'll definitely take care of it. Other questions or things?
There was a question about whether the recorded video will be sent out by email. Angie, is that that would actually be a question for Angie.
Yeah, so I was actually typing, but I'll just say, yes, so everyone's going to get a follow-up email that includes the link to the recording and also that shared folder with the resources and experiment files that we talked about today. And you'll also get a certificate of attendance as well. And I did put in the chat where you can find the recording on our website once the edits are done.
Yeah, let me, that's a good thing. Let's take a look here. Let me exit full screen. And I think, or actually, let me just exit the slideshow. I think that's fine now.
While you're doing that, there was one question that Robert had in the chat that I answered in the chat, but I think it's good to actually kind of put it out there too. He said, well, what happens if I'm using a food that, do I have to worry about it absorbing at 595 and interfering with the Bradford assay?
Oh, that's a good question.
For the most part, there's not a ton of stuff in most foods that you see that will absorb at that. But what you could also do, right, is just do a background check of that food. You dilute it just in water, put it in a spec, see how much is going on in that 595 range, and you'd be able to subtract that out of your other readings would be the way to handle that. And that is one of the things that does actually make Bradford a much better tool at diagnosing food than the protein content in food than some of the things that do get broadly used. Because some of the things that get broadly used in the food industry just measure nitrogen and use that as a proxy for protein. And this is why we had that giant melamine fraud event, I think what, in 2017 or something like that, was because that adds a whole bunch of nitrogen to it. But if you were to run a Bradford, you wouldn't see protein there because it wouldn't pick it up. So anybody that wants to use that as a context, I will drop a paper about that and the difference between those assays in the chat for you.
Yeah, any other questions? I think we've got like a few minutes left. No? Well, I just want to say thank you to everyone for coming. Just thank you a lot for attending. And if you have any questions or need anything, just feel free to get in touch. Once again, you can email biology@vernier.com. You can get in touch with us in chat. Chat is a really great place to go. If you go to chat on our website, you can just search for Sarah and I will be there. Oh, yeah, you've got that. Someone wants a link to the paper there, Damon.
I did drop it. Let me drop it one more time. Oh, that's why I only dropped it to Robert. Sorry about that, everybody. Let me drop that to everyone.
And I can include it in the folder of resources that we're providing as well.
Great. Well, thank you so much.
Thanks for attending, everybody.
Thanks, everybody.